So we're speaking with Kevin McKernan, the Chief Scientific Officer of Medicinal Genomics, a Massachusetts-based company, which a few years ago, I believe in 2011, sequenced the cannabis genome, or a particular cannabis strain.
Yes, that's right.
This would seem to have some significant implications for the cannabis industry as a whole. We'd like to explore that with you. So maybe you could explain a little bit of what we mean by sequencing the cannabis genome.
Sure. This was in 2011, and the tools we had to sequence back then were still evolving very, very quickly, but we were able to get a very draft version of this genome sequenced. And what this is reading every letter in the genome. And really in any cannabis sample, as many know, there's two genomes. There's really the mother and the father genome. The plant is known to be diploid, so it's got 20 chromosomes and one copy of each from mother and father. There's some chloroplast, some mitochondrial genome in there as well, but we want to read all of those letters so we can begin to build a map of all of the genetics that might predict cannabinoid expression, terpene expression, maybe even flavonoid expression, and as it moves into hemp, maybe some of the genes that are governing either seed size and oil and fiber. So to read the entire genome, it's about 1 billion bases long. It's 1 billion letters of genetic code. In the process with the technology we had, we probably only got about 400 to 500 megabases of really nicely aligned sequence. Now that seems like you've only gotten about half of the genome. That's probably true. There's a lot of repetitive nature in plant genomes. They have copies of things that are identical scattered throughout them, and those end up, when you sequence them, it's kind of like putting together a big jigsaw puzzle, and those are like all the pieces that look the same, and sometimes you don't know exactly where they go, and so they're in the sequence, but they get kind of left as ambiguous when you put all this data together. But we do have is really nice scaffolds of some of the genes everyone's very attentive to, like THC synthase is one that is there's really nice sequence coverage over those, CBDA synthase, and some of the genes that are governing cannabinoids.
When you say synthase, you're talking about the kind of the precursor gene for what will become CBD, or is an enzyme involved in making that
there's
happen?
an enzyme involved that so the DNA codes for a protein, and that protein folds into a little enzyme that folds the precursor molecule into either THC, and there's another gene called CBDA synthase, and the A is for the acid form because it makes THCA and CBDA before in the plant form. But that's taking a cannabigerol precursor, and there's a little, if you look at cannabigerol, it's like a ring structure, and it's got a long tail on it that it kind of folds and wraps into two more cyclical groups that either makes THC or one more cyclical group and makes CBD. And there's two different genes responsible for folding that precursor two different ways, and they're actually in competition for the precursor. So we believe, and many others before us have actually done this work to demonstrate that DNA variants in those genes have some predictive capacity on how much THC it's going to make. So there's been some publications showing mutations in key areas of the gene, and this is known as a FAD binding domain, a very technical term for a catalytic core of the enzyme that folds those molecules. And when there's DNA variants in there, it does a slower job at it. Sometimes it doesn't do any job at it. So we believe this is kind of a rheostat at how much THC is produced depending on where these variants are in the gene. Mm-hmm. And if we can get a better picture of this, we can begin to predict these chemotypes of the plant at a seedling stage without having to fully grow them out. Now, the environment's always going to play a role in this dance as well, but we're just trying to get a really firm picture of the genetics so that we can have some understanding of the capacity of the plant. If we know the plant's going to make all CBD, you might put that in a different grow room, or you might do something different with it, breed it with something else where you're trying to bring CBD in. But if you know it's going to be a really strong THC strain, that perhaps gives you a different direction to go with it.
Mm-hmm. So you've touched on some of the implications of this genetic science for cultivators, right, for breeders possibly, or for people who are trying to fine-tune particular strains, yes, for particular medicinal properties. Get into this a little bit more. How will this really be, this kind of knowledge, other than pure science, how it will be applicable for growers or for the cannabis industry as a whole?
So where we've seen this take foothold in other marketplaces is with a process known as marker-assisted selection. I'm a much bigger fan of that type of breeding than, let's say, let's go monkey around with the genes. We don't really understand the whole genome yet. So the GMO stuff, I think, is a little bit arrogant right now, and maybe even ever, depending on how much we ever get to know about this genome. But what people do tend to do with sequence information and breeding is they use the DNA markers to track the traits. And it's just kind of a measuring tool. Instead of measuring the chemotypes as they grow out, which is a great way, I don't think that's ever going to go away, but if you want to measure what chemotypes you might get at the first sign of the leaf, there's information to be had there that can tell you, okay, here's the terpene profile we think it's going to have. Now we don't know the markers that do that today, but this is what they've done in other very valuable crops, is that they get an understanding of maybe 100,000 to 1 million of these different single letter changes in the genomes, and they track those in all the plants. And they use those as sort of a proxy for, okay, all of these changes over here tend to track with terpinolene, and these ones all tend to track with caryophyllene, and these ones tend to track with maybe pinene. And so we know that those markers are predictive that this plant's going to be, maybe a myrcene dominant indica, and this one's going to be a pinene, or something that's more Jack Herrera-like with the terpinolene. So those markers, we think, at a hole punch, we could distill. That you can get a hole punch of the leaf. All you need is about 30 nanograms of DNA to do this. This is 30 billionths of a gram of DNA that you can get this information from, and it will give you maybe 100,000 to 1 million different data points on the strain. And ultimately, there'll be databases that will help you correlate that, all right, this pattern is from a plant that's on this part of the phylogenetic tree, and we know that it tends to make these types of terpenes. And we're going to track those SNPs. And they're called single nucleotide polymorphisms. So there's a lingo here where people call them SNPs. Once we have that, we can then make some more informed decisions about, okay, we have a wonderful plant over here making some cannabigerol and some cannabichromene, and we really want to bring in limonene to this. And what do we cross it with to make that happen in the fastest way possible? So we think it becomes just a tool set to help guide breeding. And this is this process called marker-assisted selection is simply a fancy term for we're going to measure this with something other than photography to perhaps guide where we go with the breeding.
So medicinal genomics has actually been gene sequencing several different strains, many different samples.
We have, yes.
And what have you found in terms of diversity or lack thereof, and how has prohibition affected that?
So it's a fascinating question. There's certainly signs of there being a genetic bottleneck toward high THC plants, but at the same time of that happening, there's been a tremendous amount of interbreeding that's gone on, that if you were to compare the variation we might see in the drug type plants next to those that we see in the plants that have had less prohibition on them, like hemp plants. Hemp plants are registered, and there's a process in some countries to actually legally grow those. You actually see far more genetic diversity in the drug types, the ones that are underground. And although they might have more THC in them, there has been perhaps a lot more exchange to find different terpenes or to breed different traits into them. And I've always found that very fascinating, that the ones that are a little bit more upboard and leveraging perhaps the registries and the patent systems actually have less genetic diversity than what we're finding in the markets that are out here.
So let's talk a little bit about the implications for the medical use of marijuana. Personalized medicine is a buzzword today within the cannabis therapeutics world. And in terms of what that means, I think on the ground for patients are sort of groping in the dark to find what strain might work best for them, what helps them the most, maybe what ratio of CBD to THC is going to be their entry point into utilizing cannabis medicinally. And that always comes down to, well, it's about that person, how that person is going to relate to a particular strain or the plant in general. But I think what you're talking about with the science of genetics, it sort of takes this idea of personalized medicine to a whole different level. So and I know also that Cortigen, your sister company, has been working on the human side of the genome. Right. So how do the two come together, and what does this mean for personalizing medicinal cannabis?
So I'm really excited about this topic because we, on the flip side of our business, we're sequencing 700 to 800 patients a month to try to find variants that help dosing patients with different drugs. Sometimes it does implicate that you might benefit if you go straight to CBD. Like the classic cases, sodium channel 1 mutations are Dravet syndrome, and they are the class of patients that are responding very well, not just anecdotally on the internet, but even in the FDA-based trials. You can't deny this, that they're having a real positive effect from Epidiolex that they're testing this on.
Just being about CBD or CBD-rich product is helping these particular children with epilepsy in terms of Dravet's, but there are many different epileptic diseases.
So we tend to sequence sequence 500 genes in that for epilepsy patients because it's a long tail. And this is really the lesson of personalized medicine. And it's kind of a lesson of the FDA is that they're not designed for personalized medicine. They're designed for one-size-fits-all drugs, that aspirins and things that just hit everybody with the same thing. Those are gone. There are no more of those. There's very few of them. And it's pretty clear now that the reason drugs are failing in the FDA and the reason the costs are going up is that they're trying to apply this model of blockbuster drugs to shove it through the FDA. What we're really seeing in cancer and all these other fields is that everyone's genome we differ by probably 4 million variants in our own genome. And so what you might describe as chronic pain could be a completely different molecular mechanism if I have chronic pain. And we begin to understand that when we dive in and sequence people's genomes, that, oh yeah, you have chronic pain because you have TRP receptors that are off, and I have chronic pain because I have a TRAP1 variant. And those might require different mechanisms for dosing. It might require different cannabinoids. And so I'm very excited about bringing those two fields together because we can see that personalized medicine, there's only one market in the country that I think is growing faster than personalized medicine, and it's cannabis. And cannabis is this market that has what looks to be about to become the largest open source set of pharmaceutical drugs that grow to high concentrations in plants that are weeds. I mean, this is a total revolution in medicine that's about to become over-the-counter for physicians, that if we can line up the right patient variants with the right cannabinoid and terpene profiles, the cost of healthcare is just going to go right down, and we can do what I like to recall it as. It's almost like a technological leapfrogging event, right? Mm-hmm. You see these cases where two different technologies, kind of the two different tsunamis that come in together, and they create kind of a perfect storm of a wave.
And the two different technologies now are on the.
I think it's the personalized medicine side, knowing the patient's genome, and then also knowing the entire cannabinoid profile of these plants. And the reason I'm fairly focused and myopic on the cannabinoids is because it's really not that myopic at all. There's just a whole portfolio of these things, and they all grow in the best vector you can imagine. I mean, there's certainly a lot of interest in ripping these genes out and putting them into yeast and see if you can keep up with the plant. Good luck.
But that's not your focus.
No, I think that's harder. I think it's a hard job to do, and the plant's very good at it, and it grows. We have all this infrastructure to grow this plant, and what you do find in some places of the pharmaceutical industry, they want to put their pathways into plants like this, because they become medicinal factories for them. And we have a science of growing this plant that's quite mature and produces tons of this stuff. So I'm actually quite optimistic about that side of the equation being done just perfectly today. But to bring the two together, you do see these disruptions happen in other markets that I'm always very attentive to, that you see things like new batteries that are coming out these days can now drive things on the ground that are gyroscopic scooters now, and cars like Tesla, and then there's solar panel technologies. And when those things come together, you see third worlds skipping landlines right now, right? They're not installing landlines, they're going right to Nokia cell phones, right? They're not going to build central banks because they have cryptocurrencies or M-Pesa-like currencies that are happening in Kenya. The same thing is going to happen, I think, in medicine with the FDA. The FDA is one form of doing medicine, but it can't scale with the innovation that's out there. And the innovation I see really happening in medicine is at this level, is the level of trying to get cannabinoids to the right patients. And that model is much more democratized. It's much more individualistic. It's much more focused on and respectful of each patient being unique. And we're losing that in the healthcare system. The healthcare system is getting very one size fits all.
So in a sense, what you're talking about is kind of eliminating the crapshoot. If you've got a drug, and you've got a person with a certain genetic constitution, and you're throwing CBD at an epileptic kid, why does it stick with some kids? Why does it work so amazingly, and why doesn't it work with others? So you would be able to determine this in advance.
We're starting to see some signals on this, and this isn't any coordinated effort necessarily with FDA or with GW. Because we sequence so many patients at Cortigen, we happen to have a lot of patients that are in those trials, and they tell us. And when we take their data and compare the responders to the non-responders, we can see that there's a pattern. They're starting to show a pattern of the responders have variants in there's like 4 particular variants that we've been showcasing that they have. And they're, interestingly enough, one's in a drug metabolism gene known as a CYP2C9 gene. It's a gene involved in the metabolism of CBD. The other 3 genes that are showing some predictive power here are sodium channel genes that we know anandamide interacts with. So there's a thesis here that kind of makes sense. And the reason it's important is some of those patients, CBD isn't their molecule. They get 200 to 400%, for small, like 15% of the kids get worse. And when seizure patients like Dravet patients get worse, it's expensively worse. It's like they get intubated, they end up in the ER. Sometimes they get status epilepticus. And oftentimes you hear anecdotally, some of those parents switch them to THC or THCA and they get better. So there's different mechanisms of causing seizures, and I think as we understand those mechanisms from a genetic level, we get more intelligent about which cannabinoids to hand them. We don't know the answer to how to mix and match that perfectly today. That's kind of the vision, that's where we want to go. But I think what we're going to see over time is that puzzle is going to slowly fill in piece by piece, publication by publication, that these variants are very predictive of success on this class of patients, and this class of patients really needs maybe a THCA, which is a completely different molecular pathway.
It's very exciting. Presumably that can be applicable to other conditions, and not just the epilepsy, but...
I think so, yeah. Well, I think chronic pain is one. So we published a paper on the chronic pain front, and some of these patients were very responsive to antioxidants, and we all know that some of the best lipid-soluble antioxidants in the world are cannabinoids. So these patients didn't happen to jump on that, but the fact that they were responsive to N-acetylcysteine, which is more of a not as much of a lipid-soluble antioxidant, so there could be some improvement there. That's very exciting to us because this variant is in 1% to 2% of the population. It's in a gene known as TRAP1 that has some implications in cancer, but it's never really been pinned to chronic pain or chronic fatigue until we were through about 1,000 patients, and we summed up all the patients that had chronic pain and chronic fatigue, and bang, a big signal pops out saying TRAP1. And when that gene's broken, the hypothesis is it's making lots of reactive oxygen species. So your body's churning through energy and creating all the side products of energy consumption, but it's not actually functional. And that's creating, we think, the pain and the fatigue, is that this enzyme's broken, it's eating up all the ATP. And so we're really hopeful that will translate into maybe making a dent in this opiate problem. Right, these people are just getting handed opiates, and it probably isn't really addressing the core issue of creating lots of ROS. It's just dulling the pain that's coming from it. And we've seen where that's gone. It's just gone into a complete opiate epidemic right now. So we're hopeful that some of the sequencing will come in and objectify what is a subjective problem for a physician. Patient shows up, says, I have pain. Sure you do. Then they have to really monitor how much dosage they give them and whether it becomes chronic, whether they get GI issues with the opiates. But if you had a molecular marker that said, no, these are the patients that actually have a broken gene that we know responds with antioxidants, you can suddenly change that conversation to, we should be considering cannabinoids for this. And I'm hopeful that's going to happen in autism and mitochondrial disease. We'll probably see it in Parkinson's and Alzheimer's. A whole host of diseases that you can see on the Project CBD website that are playing a role. And they probably all have different molecular mechanisms as to how CBD is benefiting those things.
Well, I think medicinal genomics is changing the conversation, and it's great. It's amazing the work that you're doing. And
I
appreciate
so, yeah.
Kevin McKernan for being with us today.
Oh, thank you. Yeah, we're excited about the field.